Hi, good morning, everyone. Hope you all are doing well. So, today we will start with the new lecture on AI, machine learning, and antibiotic drug discovery. So, what we are covering today is why new antibiotics are urgently needed, how AI ML accelerates discovery across the pipeline, maybe focus little bit on protein docking generative designs and some case studies that have emerged very recently. So, we all know about the antibiotic discovery crisis. I don't have to tell you or repeat myself again over here. So, basically, we have a sharp decline in novel antibiotic approval since 1980s. Then there is rising AMR that threatens the modern medicine. Traditional pipelines are costly, slow, takes many years and decades for discovery. And you know this picture already from the previous session where you need to find an appropriate target, identify a chemical or a lead molecule which has inhibitory potential, modify the lead compound as necessary to enhance the potency, evaluate in vitro activity. So look at the activity in the lab um, through antimicrobial susceptibility tests and also cell lines, assess the activity on different cell lines as well. Then evaluate in vivo activity and toxicity, working with like mouse models, for example. And then lastly, uh, take that forward into clinical trials, phase one, phase two, phase three. Hopefully, it will pass those trials and it will be in the market. The drug will be in the market after that. So, it is a very long, tedious process. Um, and you can appreciate that it will take more than 10 years and more than 1 billion or up to $1 billion is what is needed in order to accomplish that process. So this is a new slide which you have not seen earlier, but what you can see on one side, one end, on top end is you can see when the antibiotic was deployed for use, and you can see when the resistance has emerged at the bottom in the bottom panel basically. So sometimes resistance has already emerged previously. Sometimes the resistance emerges after some years, but it is not a rocket science to know that resistance is going to emerge. Resistance is always going to emerge in one or the other way. You know, it is something that cannot be stopped. Whether you develop a drug or whether you come up with whatever novel drugs, because evolution is based or evolution is highlighted in that manner. When bacteria are evolving, when things evolve, bacteria want to survive. And according to the Darwinian theory, we all know that the fittest are the ones who survive in the environment. Just like we are surviving and we are evolving, we have the fittest microbes that are there in the environment, which will survive antibiotic pressures and all the stressors that are there in the environment, all the stresses that are there. There are all sorts of stresses that microbes are exposed to on a day-to-day basis. So you need to appreciate that because we don't think in that manner or we don't think deeply enough. There are microbes that have evolved resistance to alcohols. There are microbes that have evolved resistance to quaternary ammonium compounds that you that we have in hand wash, for example. So various types of things are there, and there are genes that govern um, or genes that confer resistance to these um, compounds. So what is AI, ML, and DL? So these are very commonly used terms. At least nowadays, you will hear a lot about AI, ML and DL. So AI is nothing but, when machines can think or act smart, like humans making decisions or solving problems. Like we solve problems in that way, the machines can solve problems. So that is nothing but that is the artificial intelligence, because it is the intelligence of these machines. It is the intelligence of these computer systems. In machine learning, we have computers that learn from data. So large amounts of data is there. Um, and just like we learn from our experience, the computers or the machines, they will learn from the data that is pumped into those machines. And there are lots of things in that as well. There are training data sets and all sorts of data are used. Deep down, an extension of the machine learning is deep learning. Okay, so it is a special type of machine learning where the computer uses layers, like we have layers in our brain in the same manner to understand the complicated patterns that are there, um, such as images or speech. And sometimes this deep learning, where it's highlighted that it is using layers, it is like the neurons that connect our brains. So sometimes, like, there is a term that is used like convoluted neural network, CNN. It is nothing but it is this, deep layers that are there, uh, that the computers are using in order to analyze, in order to understand some complicated patterns. Sometimes, in these settings, it is not known how the computer's prediction is not easy, or you will not know how the machines are doing that. In some cases, that it can be trained in a certain manner, but in other cases, it is random sometimes. So that's why there are lots of terms that are used in that context. So why AI in drug discovery? So we all know, like, we need to find promising compounds faster. Um, we need to predict which molecules are likely to work against what targets. We need to identify new targets as well, because everyone has identified the targets previously, but we need some newer targets as well in order to understand how new drugs can be targeted against those novel targets, and treatment for several diseases can be possible as a result of that. Understand behavior, so estimate how drug binds, how toxic it might be, how it behaves in the body. So that is very important. And I think from the previous lecture when we described about selective toxicity, right? We looked at the plasma half-life, we looked at the uh, uh, protein binding, we looked at the absorption. So that ADMET that is highlighted over there, that is a very common term. It is absorption, distribution, metabolism, excretion, and toxicity. That is what it stands for. Okay. So you will almost certainly come across admit everywhere. And that is what I just told you. So that is very important in order to know how a drug is going to behave, how toxic it will be, um, or how it can be distributed. As I said earlier, some drugs can penetrate certain tissues, some drugs cannot penetrate certain tissues. And using pharmacology and other properties, we can understand that better. So AI in drug discovery, it saves time and money. It cut down years of lab work and high research costs. We all know that research is not a small task. It is you are doing lots and lots of work, whether in the lab, in order to identify something. So computationally, you will be able to predict. You are able to predict something, and from that prediction, you can take it forward to test in the actual lab. So it will cut many years. So discover new uses, identify existing drugs that could treat new diseases. So, for example, drug repurposing or drug repositioning. Rather than repurposing, now the term that is used is drug repositioning. So, you're positioning a drug that was used. And there are several examples. Maybe you don't know what the examples are, and we will see through the slides that there are several examples of drugs that were developed for something and then they were applied for something else at the end. And then handle big data. So, virtually screen millions of molecules quickly and efficiently. So, all this is very computationally rich and it requires high computing power as well. So that is something that we need to appreciate as well. So in antibiotic drug discovery workflow, you have the target identification. You will do the compound screening. You might have several compounds from that screening. Then you can do what is known as lead optimization. And then preclinical and clinical stages remain essential. So that is very, very important. And AI supports every step to reduce any failures. Okay, so you can have, for example, in the picture, you can see there is a training data set with antibiotic property predictor. And then you have a predicting data set, and you can see new antibiotics that can come out of that as a result of pattern recognition or um, quantity structure activity relationship, QSAR based similarity between the drugs. And further on, you can see it is being tested in the lab environment initially, and then it will go on to preclinical and clinical stages. So data in antibiotic discovery. So all sorts of data are there. We are familiar with many of these datas. Maybe you have already applied some of these datas in your own context. We have got the genomics, the omics data that is there. In that you have got the genomics, which is what I deal with on day-to-day basis. The genomes of the organisms, assemblies of the organisms. Then we have got the proteomics. We have got the proteins, identifying which proteins are enhanced in what kind of diseases, metabolomics, uh, the metabolic pathways that are there. Then chemoinformatics, chemical biology along with information technology, for example, computer science as well. That is the branch of chemoinformatics, which provide the raw material for AI analysis. And some key public databases are highlighted underneath. So Patrick is one that utilizes bacterial genomes and pathogen data. We have got BixDB database that again works with bacterial genomes. Um, then NCBI is there. NCBI is also a database where you can have all sorts of uh, data, excuse me. Then you have got CARD. You have got the CARD database, which looks at antibiotic resistance genes and mechanisms. We have got Cleborate, for example, for Klebsiella pneumoniae. We have got what else? Many databases are there. There is antimicrobial peptide associated database for antimicrobial peptides linked with Acinetobacter or other antimicrobial peptide associated databases are also there. Um, then PubChem Campbell has compounds with which will have the chemical structure and can have bioactivity associated data. And data quality is something that matters quite a lot. We should have clean, well annotated data sets, and those are crucial for accurate ML predictions. So pre-processing of data is also very important. Without that, you can't be sure about your final result. So everything involves a pre-processing stage. Whenever you are having lots and lots of large data or big data, which is what we call pre-processing is involved, and in that pre-processing, we are trying to understand how, for example, for the genomes, I will look at the genome assembly, the fragmentation size, the number of ends that are present in that genome, the annotation of those genomes. Various factors are there, the length of the genome compared to the standard bacterium that I'm analyzing. Uh, So all sorts of things are there that need to be considered, and only then it has to move on to the next stage. We sometimes are in a hurry and we have lots and lots of data and we just move to the next stage. But reprocessing is very, very important. And that is why I'm highlighting that, clean, well annotated data sets, because only then you can make some accurate predictions. So integration powers, combining multi-omics chemical data helps AI uncover hidden drug target relationships. So if you have, proteomics data along with genomics or any other form of data, then obviously it will um, speed up the process, or it will enhance your discovery, is what it is saying. Because you can also look at, RNA-seq data is there. For example, in RNA-seq, you can understand what kind of genes are being expressed and how those genes are involved, whether in drug resistance or whether they can be used as targets for inhibition of the bacterium. So those are the things that uh, can be integrated using the multi-omics data sets. So target identification using AI. So predicting essential targets. So AI analysis, you know, AI analyzes bacterial genomes to find genes or proteins crucial for survival using features like conservation, expression, and knockout data. So conservation, we already know. When you have, I spoke about Staphylococcus or other bacteria. Like if you have a strain number, I don't know, ST50 of Staphylococcus, and then you have another Staphylococcus ST21, then those particular strains will have certain conserved regions. There are certain conserved genes. So because Staphylococcus is a gram-positive organism, one of the main targets in gram-positives is the cell wall. So in the cell wall, you can identify, okay, what region, what things can be targeted, and newer compounds, newer drugs can be developed for that. But other than that, there are also other outer membrane associated proteins, maybe purines or something like that in the gram negatives that can be targeted as well. Um, and when those things are targeted, then you can see um, a compound that can be effective against those class of microbes. Then expression is hand in hand with the gene. You have got the gene, you're looking at the expression of that particular gene, because maybe that particular gene is overexpressed. If you have an antibiotic uh, that is targeting the cell wall, for example, then maybe sub-inhibitory concentrations of those antibiotics might be doing something to the expression levels of the cell wall, because the bacteria already knows that it is under stress of that antibiotic, and it is synthesizing, you know, more of the layers, or protecting it, or coming up with alternative mechanisms by which it can protect itself. So expression data is important. Knockout data is nothing but you have the gene and you're trying to understand how important is that gene, because if you knock out that gene, then the function is lost. So, and remember that, I mean, as much as I like all these kind of things, I always like to look at a very big picture. And I give an example to anyone, professors or anyone else listening to me. I always tell them that it is not, because I work with clinicians, I work with so many diverse range of people across several disciplines, inter and cross disciplinary. My work is inter and cross disciplinary across social sciences, humanities, economics, uh, chemical biology, net zero, all sorts of fields. So, what I say is that maybe you have seen this picture already, but it's not highlighted over here. But what I'm saying is that you have an elephant in the room, right? Probably you have seen that picture of the elephant when there are five people who are touching the elephant. One is touching the trunk, another one is touching the eyes, another one is touching the tail. And they feel how the elephant is, how someone who is touching the tail is feeling like, oh, it is furry in nature. Someone who is touching the trunk is like, okay, it is rough in nature. So that is an individual perspective. But if you are solving a problem, then you need to look at the whole elephant, how big it looks, and what is the actual depth and breadth of that animal. If you want to do something. And it's the same in biology as well. You have got certain genes will play an important role, but question that needs to be asked is that gene the only gene? You know, if that gene doesn't exist, then are there any compensatory mechanisms? Is there anything else that can compensate for that kind of resistance or whatever? And you will be surprised to know that sometimes there are things. It's just like, because you don't know, you only touch the tail of the elephant, so you think it is furry in nature, but it's not like that. It is a huge. So when you're looking at the problem, you have to look at the problem in a very big sense, in a very broad sense, and that is something that um, is very, very important. Without that, you can't have major breakthroughs in science. So network-based insights, we already know, like PPI, everyone is familiar. Protein-protein interactions. Protein-protein interaction is nothing but, a single protein will not do a job. You will have sometimes interaction with multiple proteins that are part of an hierarchy. You will have proteins at a lower hierarchy and proteins that are there at a higher hierarchy that are being influenced. Um, Gene co-expression networks can reveal like weak points, and you can identify novel targets as a result of that as well. Um, So target identification using AI. So why these models? So there are certain models, and I'm not going to go very deep into these models, but I will give you very briefly what it stands for. So random forest is just like a forest. Is basically there are random forests, and um, these are, so it handles many features and identifies key predictors robustly. um, and it is based on um, certain certain data sets, certain number of data sets that it works with, or certain type of features that are grouped together, just like, in imagine individual forests but randomly. So it will utilize random features. It doesn't know what it is doing. It is like a trial and error is going on, and with the trial and error, it will work in a way that you will get a result, or you will get a prediction at the end Then support vector machines work well with high dimensional genomic data and separates essential versus non-essential genes. So support vector machines, they work again. It is related to mathematics. There is a plane, and in that particular plane, plane is nothing but a region, and in that particular region, it will um, it works well with that high dimensional genomic data, and it will separate essential and non-essential genes. So that is support vector machines. And then you have deep neural networks, which will capture complex patterns in large multi-omics data sets for better prediction accuracy. So again, in the picture, you can see you have got the training data set, you have got the predicted compounds, then you have got the neural networks that will analyze that. Then you have the high predicted score and you have the low predicted score. And based on the high predicted scores, you can test those compounds for the spectrum of activity. You might find that they have a narrow spectrum of activity. You might see that they have a broad spectrum of activity. An example of that is highlighted over there, which is abaucin, which was one of the recent antibiotics that was discovered against Acinetobacter baumannii by researchers in Canada, and it targets the lipoprotein pathway in Acinetobacter only. So it is a narrow spectrum antibiotic for Acinetobacter. There still needs to be a lot of work in done in that area. Compound screening, predicting activity. So AI predicts which compounds are likely to inhibit bacterial targets before lab testing. Computationally we can do that. You have got the QSAR, quantitative structure activity relationship models, um, related to chemical structure features to biological activity. um, and here also, for example, in QSAR you can have, for example, quinolones or fluoroquinolones. In quinolones you have got the normal structure. In fluoroquinolones, we have got the fluorine at the carbon 6 position. Um, and you can see that, for example, fluorine is something that will enhance activity. It will enhance the activity of the fluoroquinolones. So those things are important, understanding how that activity is related. Descriptor based machine learning using molecular descriptors to predict uh, potency or toxicity. For example, looking at the molecular weight or looking at, I don't know, the dosage size. Um, then you have got like deep learning, which captures complex non-linear patterns in chemical data for better prediction. And there are several platforms that are highlighted: DeepChem, ChemProp. These are Python-based tools which can be used, uh, widely used for chemical property prediction, for example. And then you have got like active learning, um, where AI suggests the most informative compounds to test next, improving efficiency. So it is constantly a learning process, is what I'm saying over here as well. You have got the data set and it just is refined further quite a lot. So compound screening, uh, model learn pattern from large data sets of chemical structures and experimental outcomes. Sometimes you have a set of compounds, and you also have experimental features related to the set of compounds as well. And features like molecular descriptors or embedding help predict activity, toxicity, and admit properties. So high scoring compounds are prioritized for lab testing, just like we saw in the previous slides. Compounds that will give a better score, they are taken further. So outcome of that is obviously it will save time and resources by focusing on the most promising compounds for wet lab validation. Yeah. Excuse me. So AI in virtual screening and docking. So traditional molecular docking, we know what molecular docking is. Its purpose is to stimulate how molecules bind to bacterial proteins. And there are tools like Autodock, Glide, um, which you can use. The process for that is you are preparing protein and ligand structures. Obviously, you have got the protein or you have got your enzyme and you have got the ligand, which is your antibiotic like meropenem or anything else. And you're then performing the docking simulations and then you're trying to score and rank based on like root mean square deviation and other parameters in order to understand, which ones are good or which ones will give a good activity. Sometimes there are other rules as well. Like there is like Lipinski rule. You might have heard of it. I'm not sure how many of you have heard, but you can read about Lipinski's rule. There are certain compounds that follow this. Mostly the compounds need to follow the Lipinski's rules, but there are certain compounds that don't follow the rules as well and are still good candidates. So aspirin is a very good example of, something that doesn't follow the Lipinski's rule. So you can read more on that. Okay, so that is important. So limitations, time consuming for large libraries, limited accuracy with flexible proteins, doesn't learn from the past data is what is the limitations. So ML enhanced docking, ML models predict binding affinity and possess faster and more accurately. Um, Tools for these are like DeepDoc, GNINA, and ML enhanced Autodoc workflows. Models are trained on known protein ligand complexes and then they learn patterns in binding interactions to prioritize promising compounds. And benefits of this is faster screening of millions of molecules, improved prediction accuracy, reduces unnecessary lab experiments as well. Then protein-protein docking is also important. As much as we know, we have got the protein ligand interaction. You have got the protein and you have got a ligand or an antibiotic, for example. But you also have like proteins that interact with each other. Actin myosin is a very common example of a protein-protein interaction. Everyone hopefully knows what is actin and myosin, right? These are muscles, um, proteins involved in the muscle contraction. And there you can see. So you can apply these methods in a variety of manner. So proteins often work together in cell like team members in a factory, understanding um, how they interact, and they help to find weak points to stop bacteria from proliferating. So purpose is to predict how two proteins physically bind to each other and why it matters. It targets essential bacterial processes or virulence factors. Sometimes, as much as antibiotics are needed, sometimes you also need antivirulence compounds. Probably you have not heard about antivirulence compounds, but there is a emerging area in that field as well. I know there are lots of antivirulence associated compound. Antivirulence compounds are compounds that will decrease the pathogenic potential of the microorganisms by targeting the virulence mechanisms. And virulence mechanisms can be anything. It can be capsule production, it can be fimbriae attachment, so or flagella synthesis. So think outside the box. There is not like only one direction to follow. And I gave you the example of the elephant for that reason, in order to think in the broad sense. So, health design molecules that can block these interactions, you know, and then insights are gained as a result of that. We visualize where proteins touch each other, identify hotspots that are critical for interaction, any active sites that are there, for example. And it helps design drugs that prevent proteins from working together, weakening the bacteria, and when bacteria become weak, you can kill them basically. So AI in PP and peptide docking. So protein-protein interactions are large and flexible, it making it hard to find the correct binding um, and pose using classical method alone. So AlphaFold Multimer is an example of AI-enhanced docking. So uses deep learning to predict multimeric protein complex structures from sequences. Then you have got deep dock employs graph neural networks to model protein surface and docking interfaces. CNN, convoluted neural networks based docking models, analyze 3D grids of protein structure to score uh, binding. Ensemble and target learning approaches combine multiple models on PDB and docking data sets for improved prediction on novel proteins. So applications are design inhibitors to block secretion systems or bacterial toxin complexes. As I said earlier to you, virulence factors. This is one virulence for factor. So secretion systems, there are all sorts of secretion systems. If you are not familiar with bacterial secretion systems, then go and find out what these secretion systems are. So bacteria have evolved to secrete certain molecules or effector molecules that can affect the bacteria around them, another bacteria around them. They can also affect target cells, for example, that surround it, human epithelial cells or whatever is there around. So try and find out what the secretion systems are doing. So inhibitors of those can be designed as well. Then bacterial toxin complexes as well, certain bacteria that um, produces toxins to inhibit those toxins. So prioritize protein-protein interactions for experimental testing as well. So you can use this and you can perform an experimental analysis to understand um, s- how effective it is. So predicting antibiotic targets, how it works is by analysis of sequence features, whether it is gene or protein data, then predict 3D structures, um, and then scores from knockout or other functional studies, machine learning models, learning uh, patterns from known essential proteins to predict new high value targets. So identifying targets that were previously unidentified. So identification of enterololin, a narrow spectrum antibiotic to selectively target harmful gut bacteria, specific strains within the Enterobacteriaceae E. coli, sparing beneficial microbes in the gut microbiome. So this is an antibiotic or this is a compound, enterololin, narrow spectrum antibiotic that was identified against certain pathogenic strains of or certain pathotypes of E. coli. So it will target only those pathotypes. Can read more on that. So benefit is reduces trial and error in wet lab studies, shorten development timelines, and focuses resources on the most promising targets. Next is drug target prediction. So predicts how likely a drug will bind to a target and estimate binding strength affinity. So how it works is by supervised machine learning models are trained on known drug protein interactions. And inputs, you have got the molecular descriptors of drugs, um, structures of those sequence features of proteins, and common models that are used are similar ones that we discussed earlier: random forest, um, support vector machine, neural networks. And the benefit is prioritizes promising drug target pairs for experimental validation, saving time and resources. De novo antibiotic design. So de novo is nothing but new, from the scratch. So AI generates entirely new chemical compounds that can inhibit bacterial targets. So generative models such as GAN, generative adversarial networks, or VAE, and autoregressive model propose novel molecular scaffolds. It explores vast chemical space, often beyond human intuition, to discover molecules not previously considered. So, how helicin is was discovered using AI-driven approaches by Stokes, Jonathan Stokes. And then you have got abaucin, which was identified through deep learning guided discovery, which was targeting Acinetobacter baumannii. And abaucin also was that kind of a drug which was, you know, it was not a known antibiotic as such, but using big data sets, using thousands and thousands of libraries, virtual screening, um, they were able to highlight these compounds. Abaucin is a chemical analog of something, I don't remember, but it is an analog of I think GPCR receptors or something like that. So that is the link. So as I said to you earlier, that you can have something else targeting something in human body, but if you modify that and if you utilize that, um, you will find that it will act against bacteria. An example of fluoroquinolone is also in the same manner. When the discovery of quinolones happened, people were working on chloroquine, which is a malarial drug, and quinolone was a byproduct of during that process. And then they discovered that you can utilize quinolones, or you can use quinolones for antibiotics for bacterial infections. Then they converted into fluoroquinolones, and fluoroquinolones had more potential. And then you have the third generation, second generation, third generation group of quinolones as well. In the first generation, you have got nalidixic acid, which was the first one, then ciprofloxacin, then norfloxacin, then gatifloxacin, and moxifloxacin. So it is an important process. It is not just like randomly you will find something. It is step by step, but you need to identify the potential in the initial stages. So helicin, you can see over here 2020, 100 plus million compounds, and in silico identification was done. Often target cell wall, DNA or protein synthesis. That is the conventional antibiotics. That is what they do. Helicin disrupts bacterial electrochemical gradient. So you can see a novel mechanism emerging over here, impairing energy production and killing cells. Active against multidrug resistant Abomanii, Clostridioides difficile, MTB. Its unique mechanism makes it less prone to existing resistance, which is good just now. Okay, so this was again like a discarded molecule. We didn't know that, okay, it can do all these wonderful things, but you can see AI machine learning, how it is helping that. So significance of this is it shows how AI can rapidly discover novel antibiotics with new mechanisms to fight drug-resistant infections. So predicting drug development, so how it works, ML models stimulate mutation trajectories and resistance emergence using sequence features, protein structures, historical resistance data, um, to forecast. And random forest, for example, can be used for classification of resistance versus susceptible variants or intermediate variants as well. SVM for sequence or structure based resistance prediction, recurrent neural network to model evolutionary mutation patterns over time, graph neural network to capture structural features of protein targets. So you can even predict what is going to happen, how quickly the resistance can develop in certain bacteria as well as a result of the features that are known. So FDA repositioning or repurposing, I already highlighted to you earlier what is repositioning. It is you are taking a drug that is used for, I don't know, treatment of blood pressure or treatment of diabetes, and you are using that for treating bacterial infection. So AI models screen large libraries of approved compounds for potential antibacterial activity, leverages known safety pharmacokinetic profiles. So we already know, no, like for certain drugs, we already know whether they are toxic, whether they are what, right? Because they are already approved, right? So that is another important thing. So it reduces the time, it reduces the effort. A new example of that, again, not highlighted over here. One of my friend, he has discovered aspartame. Everyone knows what is aspartame, right? Is just, yeah, artificial sweetener. So that is used for killing bacteria. Did you know that? Probably no one over here knows. So already the safety profile and all is known because it is, you know, kind of safe to use in humans and it is used as an artificial sweetener. But on the same side, his discovery has led to a novel area that has opened where aspartame based wound dressings are could become common if approved in the hospital setting. Okay, so many things are there and many discoveries are going on. And I know Jonathan who discovered ABOS and I know the other person. So I interact with these people, and obviously, as I said, because I have the network with clinicians and other people, I bring a different angle towards what they are doing. They are focusing on the discovery and other side of things. Then I say, okay, from the pharmacology and other side of things, other things need to be considered as well. So we already know it will shorten development timelines and cost and other things. So example of that over here is highlighted. Fendiline, which is repurposed, you know, drug which was used for heart arrhythmia, and then it is used for, you know, lipoprotein trafficking to kill antibiotic resistant A. baumannii. So probably in the same mechanism, targeting the LOL-E and that pathway, lipoprotein pathway, because gram-negative bacteria, I told you already, lipopolysaccharide is there. But certain gram-negative bacteria, they have a specialized transport system as well. So that is what you need to consider. It is E. coli is not Acinetobacter, and Acinetobacter is not Pseudomonas or Klebsiella. No, everything is different. Certain bacteria will have certain features, but some features are very, very different in which the pathways and machineries and the mechanisms are a lot different. So toxicity, estimate prediction, again, I don't need to tell you what is all this absorption, distribution, metabolism, excretion, and toxicity to ensure safety. You can't have a drug in your body all the time. It has to be excreted. It has to be certain half-life needs to be there. If infection is there in a body, or if there is a bacteremia or septicemia in patients, sepsis in a child, for example, or immunocompromised patient, then you need to have a drug that will retain that half-life as well, that it's not degraded quickly. So many factors are there that needs to be considered. It crosses the blood-brain barrier as well. If he has meningitis. So many things. There are drugs that will not cross those barriers. So you need to consider all the different features. So machine learning models analyze molecular structure, chemical properties. You predict how compound is absorbed, distributed, and eliminated in the body, as well as potential toxicity. And then common models again, same models are there: random forest, SVM, deep neural networks, sometimes combined with ensemble approaches for better accuracy. Data. So use AI to analyze large-scale biological data sets, proteomics, and other branches of omics pathways, uncovering resistance mechanisms and prioritizing works. AI ML models detect pattern and correlations in omics datasets that are too complex for humans to identify. And examples of that is, sorry, excuse me, predict essential bacterial genes from genomic data, identify druggable proteins from proteomic or uh, metabolomic profiles, map regulatory networks from transcriptomic datasets. So an example of this is one of my students, he worked on, AI ML associated novel target identification for Acinetobacter baumannii. Everyone knows Acinetobacter is my favorite organism. So international clone 2 is the main clone of Acinetobacter that is expanding in the world. So use the big data set of that, then did the annotation, then looked at what proteins are enhanced, which proteins are important, which pathways were coming out from that. He used like um, several databases as well, like you use like cake pathway and other PPI interaction tools as well, and identified a specific protein that is not identified previously, and then looked at databases like Campbell and other natural product databases are also there. That is also an important way to go ahead to identify new compounds. So identified a compound from there, showing the potential of that compound to target that novel protein that we had discovered. So that is just a very brief summary of what we had done. So we are hoping to publish that paper sometime. So combining AI with CRISPR and synthetic biology, leverage AI to guide genome editing and synthetic biology approaches for antibiotic discovery, bacterial engineering, CRISPR guided target validation. AI predicts essential genes and optimal CRISPR editing sites to knock out or modify bacterial genes. Synthetic biology design, AI models suggest novel pathways for producing antimicrobial compounds or optimizing metabolic routes. So, in certain bacteria, you will know that there are certain biosynthetic pathways. There are lysopeptides, there are different clusters in bacteria, and these clusters can be used for the production of new compounds as well. So you need to know how, what is the biosynthetic pathway basically, or how the drug can be extracted. So synthetic biology is very important over there. Integration, so combining omics, structural modeling, functional predictions to design precise interventions. Applications are, identifying essential bacterial genes as antibiotic targets, engineer bacteria to produce novel antibiotic compounds, which is what we need. Um, Predict genetic modifications to reduce resistance or improve efficacy. And the benefit of that is, speed ups experimental design, reduces trial and error, enables targeted and efficient development of antibiotic. So cloud and big data platforms are, everyone has heard of cloud storage, Amazon cloud storage and Microsoft and all these. big companies are there. So it is for storage and processing and analysis of massive biological, chemical data sets to accelerate AI-driven antibiotic research. So data storage, we have got centralized repositories for genomic, proteomics, and all sorts of data. Then we have got high performance computing, which supports large scale AI ML model training and simulations. So you need that high performance. Without high performance computing, you can't do. You can't do it on the normal computer. On normal computer, limited things you will be able to do. But other than that, you need the high performance clusters that are there that are screening these data sets and integration. So combines diverse data sets for holistic analysis. And I've already told you the examples of what systems are available. So speeds up computations, facilitates collaboration and data sharing across research groups. You can be working here, I can be working there, and we can be working together on the things as well. Um, Makes AI driven discovery scalable and reproducible. So that is what happens. So data limitations, so biases in data sets and limited um, label data for rare or emerging pathogens. Obviously, if there is something that we don't know, then we don't have the means. So there can be biases associated with that if the data set is small. Model interpretability, lack of interpretability. So there are black boxes. As I said to you earlier, sometimes we don't know how predictions are made, is a question mark. Experimental integration, and there in predictions you can't do reverse engineering to understand how the predictions are made. It is very, very difficult. You just don't know over there. Randomly it is going on. We don't know what the model is using. For some models, yes, we know. Others we don't know. Experimental integration, bridging computational predictions with wet lab can be time consuming, costly. Ethical IP concerns, data privacy, ownership, intellectual property of AI generated drug candidates. One of my friends, Cesar, Professor Cesar from US, he has isolated antimicrobial peptides from the ancient Neanderthals and other humans, right? So over there, the concern again is of IP. Who owns the candidate over there? That man over there, or we who discovered or are rediscovering that to the analysis of um, the genomes that are there. So there are various things to consider. So overall impact. So while AI accelerates discovery, these challenges must be addressed to ensure robust, reproducible, and ethical antibiotic development. So opportunity for researchers, there are lots. You identify novel bacterial cancer targets using genomic and transcriptomic data, explore regulatory network and pathways to multiomics integration, predict gene essentiality, expression changes, mutations linked to resistance, reposition existing molecules, and analyze their effects on gene and protein networks. Combine AI with CRISPR and synthetic biology for precise functional validation. Utilize cloud-based data platforms for large-scale omics analysis collaborative works. And real world impact is there in the way of faster drug discovery, novel therapeutics, drugs that are repositioned from something else into antibacterial or antibacterial drugs that become anti-cancer, anti-diabetic drugs, um, resistance and therapy management, then precision medicine as well. Precision medicine is nothing but tailored. It is tailored to that specific disease. That is called as precision medicine. And then resource efficiency to reduce cost and wet lab workload through in silico screening uh, and multiomics analysis, AI driven predictions. So future trends are moving in the direction of like hybrid workflows, combining MD simulations with ML for accurate scoring of drug target interaction, precise antibiotics and AMR surveillance. Sometimes we do predictive modeling. I work with another colleague of mine in Aberdeen where we are predicting the future, what is going to happen, um, how much the drug-resistant Acinetobacter or Klebsiella pneumoniae is going to move ahead, what kind of infections it is going to cause. Today, it is causing maybe 100 bloodstream infections in a hospital. I know in the UK, in several hospitals, I already have the data set that I've curated. I know this is there. So I can make a prediction in the next 50 years. This is what is going to happen. Another time, one of my other student has made a prediction of the foodborne illnesses of Campylobacter, how much it is going to increase by 2050, and what is the economic cost, what is the economic burden on the economy as a result of that. So that is predictive modeling. That is another branch. Generative molecules, to create compounds with uh, inbuilt resistance avoidance features is not easy. All these things are whatever I'm saying, they are not easy things to do. Remember that. And there are so many challenges. You need to do it in a very step-by-step manner, but you need to identify the potential at the initial stages. If you are able to do that, then you'll do well. And then integration with multi-omics synthetic biology, combining AI prediction with gene editing, transcriptomics, metabolic engineering for smarter drug design, design smarter drugs. We have smart cities. You can also have smart drugs as well in the future. Maybe you will be someone who will design smart drugs in that domain. All right, so that's it from me. I think there's break time now, so we can go and have a break.